All right, welcome to the second part of Chapter 10, Cellular Reproduction. We're going to work through comparing asexual to sexual reproduction, and then we'll look at asexual cellular reproduction in eukaryotic organisms. We'll talk about the cell cycle and the stages of interphase, the stages of mitosis, and we'll look at how cells divide during cytokinesis, and then how mitosis is controlled. Remember from the previous lecture, and from Chapter 1, way back in the beginning, reproduction is one of the properties that all living things have, and it's essential because you need to continue the species. And without reproduction, that's not going to happen. We looked at asexual reproduction a lot last time when we talked about binary fission in bacterial cells. But there's also asexual reproduction going on in eukaryotic organisms, including multicellular organisms like you. The advantages of asexual reproduction is you don't need two parents. You only need a single parent or a single parent cell. The downside is you don't get very much variation. And variation is important because with variation you get diversity. And with diversity you get strength. The strongest species are the ones that are the most diverse. That's why humans are so successful, because we have a huge gene pool with a tremendous different number of combinations of genes that can be found in an individual, and that allows us to adapt very readily to almost any ecosystem. All organisms, whether they're single-celled or multicellular, have some form of asexual reproduction going on, and we typically think of binary fission and mitosis as the two main mechanisms. And we'll look at mitosis specifically today. We already looked at binary fission last time. Sexual reproduction, you got to have two parents, but you got a lot better chance of variation because you're getting DNA from two different sources. Only eukaryotic organisms reproduce sexually, although some prokaryotes can exchange some DNA. We talked about plasmids, but they're not on the chromosome, so it's not really, like you're fusing chromosomes from two different cells. And then we have to use a different process to make gametes. The sperm and the egg have half the chromosomes that the zygote has, which is the fertilized egg, that then develops into the multicellular embryo. And so we need a process to reduce the chromosome number, and that's meiosis, which will be covered in another lecture. Eukaryotic cells also divide asexually, and there's also binary fission. There are some that do binary fission, like some protozoans. There are others that can regenerate by, here's a hydra, a green hydra that's growing, that's budding, growing a bud off of the side. Here's a planarian, a flatworm. I used to do this activity long ago where you'd take planarians and you cut them in half with a razor blade, and each half would regrow the missing parts. And so regeneration is a way to, or by fragmentation, take a parent organism, it gets chopped into pieces, and then each piece can develop into a new organism. And then there's sporulation, which we see especially common in the fungi. And if you've ever seen fuzzy bread, the fuzzy part that you're seeing are these asexual fruiting bodies that are developing spores that can then germinate later into new organisms. Eukaryotic, the reason most of the time, though, we don't use binary fission in eukaryotic organisms is because if you're multicellular, you're likely to have multiple chromosomes. Our eukaryotic chromosomes are paired, and there are many. Humans have 46, 23 pairs. And when you have that many chromosomes, you can't use a simple process like binary fission to replicate, and that's why we use mitosis. When we look at, if we were to take any of your cells, they have what's called a cell cycle, or an ordered series of events during the life of the cell. And there are two stages to this. The first stage is interphase. Cells spend about 90% of their time in interphase. Some cells never leave interphase. Cells that are not gonna reproduce, like your muscle cells and your nerve cells. That's why when someone has damage to their nervous system or to muscles, it's usually permanent, because those cells can't replace themselves. They can't fix any damage. Interphase is normal cell growth, but there will come a point where if the cell is going to reproduce, then it has to start to prepare for division, And then once it's ready to divide, then it goes into the mitotic phase. And this is when the single parent cell is going to split into two identical daughter cells, and the very end of that is called cytokinesis, which is the final splitting. There are three stages or phases to interphase, G1, S, and G2. The first one, G1, first gap phase, this is normal cell growth. So at the beginning, when a cell first comes out of mitosis, it's going to be small. And during G1, it's going to grow, and it's going to mature, and it's going to take on whatever its final role in the organism is, if it's in a multicellular organism. And some cells, again, aren't going to reproduce, muscle and nerve cells, or older cells that aren't able to reproduce anymore. They're not dead yet, but they're called senescent because they're not going to reproduce anymore. And oftentimes you'll see people create this G0 phase where those cells that are never going to reproduce are not really in G1 anymore. For me, I don't know that it's necessary, but you're going to see this in the reading, so I want you to be aware. A key stage of interphase is the S phase. S stands for synthesis, and this is when we're going to take all those chromosomes and we're going to duplicate them. We're going to make copies of them. And the copies are going to stay joined together at something called the centromere, which is somewhere in the middle. And again, these are identical chromosomes to each other, and what's going to happen is when we get it to the end of mitosis, we want one of these in each new daughter cell. It's going to be important that one copy of each chromosome goes to each new daughter cell so that each daughter cell has the full complement of the organism's genome. And this is showing you an electron micrograph of sister chromatids. They're highly condensed at this stage, and the centromere is here in the middle holding them together. The final part of interphase is G2. And this is where the cell is going to get bigger, and it's going to start stockpiling all the monomers and polymers and extra membrane and extra organelles that it's going to be required for one cell to divide into two. This is also when the cytoskeleton is going to break down, because if we want the two cells to divide, we don't want the cytoskeleton there still holding things together. And if whatever energy was utilized by the cell in S is going to be replenished so that the cell has surplus energy to make it through the M phase. The mitotic phase, when the cell is actually going to divide its chromosomes and form two daughter cells, is kind of split into two parts. There's karyokinesis, which is the four major steps of mitosis, when we're going to break down the nucleus, separate the chromosomes, and then reform a nucleus around both sets of chromosomes in the new cells. And then finally cytokinesis, when we physically separate the two cells from each other. The thing to realize about mitosis, we're going to talk about these stages, prophase, metaphase, anaphase, telophase. I included prometaphase, but I typically don't consider it to be a separate phase, but some scientists do, and the author of our textbook did, so I want you to be familiar with the term. But the truth to me is that all the things happening in prometaphase really start in prophase and end in metaphase. So it's not, to me, it seems like an unnecessary step. But when you have these steps, it's not like you're in prophase and then click, you bump into metaphase, and then metaphase and click, you're in anaphase. It's a gradual change. It's like when you were born, you were a small baby, right? And one day you didn't just poof turn into a toddler, and then another day later, poof, you turned into a small child, and then a bigger child, and then it's a slow, gradual change. And the same thing is true of mitosis. This is also when the cell is subject to damage. So mitosis actually happens pretty fast, because this is when the cell is susceptible to environmental factors that could kill it. Okay. Now cytokinesis, even though we say it's at the end of telophase, that's when it stops. It's probably going to start as early as anaphase, so that the cells are slowly dividing from each other while we finish up the mitosis stages, and then we get ready to split. A real key structure in mitosis is the mitotic spindle. And the purpose of the spindle is to make sure that both copies of each chromosome, what we call sister chromatids at this point, don't end up in the same daughter cell. Because then you'd have too much information in one cell and not enough in the other. And it's composed of microtubules. And what ends up happening is these microtubules are going to bind to the sister chromatids. And then when they separate, the spindle fibers are going to shorten, and it's going to physically pull the two sister chromosomes. Now we call them chromosomes when they separate. Going to pull them apart from each other. Way back in an earlier chapter, we talked about the parts of eukaryotic cells, and we talked about centrosomes being made up of a pair of centrioles that are at 90-degree angles with each other. And it's the centrosome and centrioles that are responsible for building the spindle fibers, some of which are going to be used to pull the sister chromatids apart. Others are going to lengthen the cell. And so this is a key structure in mitosis to ensure that one copy of each chromosome goes to each new daughter cell. Now, the first part of mitosis is prophase. And in prophase, this is where we start to build a spindle. We want to separate the chromosomes, so we're going to have to break down the nuclear membrane. And when we pull the sister chromatids apart, the chromosomes apart, we can't have them long, thin strands of chromatin. We've got to supercondense them, supercoil them. And we coil them up so that they're much, much shorter. And now they're so short you can physically see them with a light microscope. These are light microscope images of an animal cell and a plant cell. And you can see how the chromosomes are visible now because they've been supercoiled. Okay, so these are the three things that are happening during prophase. In metaphase, our spindle is fully formed, and the sister chromatids are going to line up along the middle, what's called the equatorial plate or the metaphase plate, right smack in the middle of the spindle. And each pair is going to be attached to a separate spindle fiber. It's going to be attached with kinetochore proteins that attach the spindle fibers to the proteins of the centromere. And once they're firmly attached, and all the different sister chromatids are separated from each other on a different strand or a different microtubule, then we're ready to go to the next phase. Okay, and there are cohesion proteins that are holding these sister chromatids together until it's time for them to separate. Anaphase, the cohesion proteins break down, and so now we can separate the two sister chromatids. We call them chromosomes at this point. And what's going to happen is microtubules over here at the centrosome centriole end are going to start to shorten, and as they're shortened, it's going to physically pull that microtubule towards the end, and it's dragging that chromosome with it. And so we're physically pulling the two chromosomes apart. At the same time, other microtubules are going to get longer and push the cell, so the cell begins to elongate and get bigger. Finally, we get to telophase. In telophase, our chromosomes have reached the poles. The microtubules of the spindle start to break down. New nuclear envelope starts to form. The spindles start to unwind, and we're continuing to get ready for cytokinesis. And you can see in an animal cell, you can see how there's what's called a cleavage furrow because there's a ring of contractile fibers. They're slowly pinching those two cells in half. In plant cells, you can see this thin cell plate developing right in the middle between the cells called the plasmalemma. These are composed of vacuoles that are loaded with cell wall material. And as more and more of them accumulate there, they fuse. And when they fuse, now you've got cell wall material on one side and you've got cell membrane on the other. And so the reason it's different is, see, plant cells are likely to stay attached to each other after mitosis to make plant tissues, whereas animal cells are likely to be separated. And if they are going to be attached, it's junctions that are going to hold them together. So the two different mechanisms for animal and plant cells are really important. So let's pop in a discussion question and have you look at this cell, and I'd like you to think about what mitotic stage the cell is in. You can pause the video if you want, although this is a pretty quick one. You probably already have an answer. And you should have answered C, this is anaphase. Okay. Now, cell division needs to be controlled. Not all cells are going to divide and develop at the same rate. Some cells are never going to divide and develop. Once once you're born, your muscle cells get bigger, but you don't get any more of them. Same way with your nerve cells. So how is division controlled? Well, first you got to have enough nutrients for growth and division. If you don't have enough nutrients, then it's not going to happen. Sometimes it's because there's a loss or death of adjacent cells. You think about if you cut yourself, you've got to fill in that gap. Or we remember when you made the slide of the cheek scraping, right? So you remove some cells, kind of like they're removing cells with a spatula here. The cells on either side then are going to get the message that they have to grow to fill in the gap. Now, cells need some things in order to grow, and one of those main things are growth factors. Without growth factors, growth isn't going to happen. And so other cells that are going to produce these growth factors that allow these cells in the middle to divide. Cells also have something called density dependence inhibition. And what that means is when cells are growing together to fill in a gap, or if we add some cells in media onto a surface, those cells will replicate until there's a solid layer of cells, and then they'll stop. Normal cells won't form a pile or a lump. They form a single layer. Okay. There's also anchorage dependence. In order for these cells to divide, they have to be anchored to a surface. See, cells that slough off, we don't want those to replicate in the blood vessels, because blood vessels are often, the capillaries are so small that only one cell at a time can pass through. And if cells were replicating, they would plug up capillaries and cells would die. So in order for all of this to be controlled, there are going to be proteins at various checkpoints that are going to regulate whether or not the cell can go into the next phase, check and make sure nothing's wrong. If something's wrong, the cell's going to be destroyed. Okay, and there are four major checkpoints. Your book talks about three of them. I added one. The first is a checkpoint at G1. We get a message that says this cell needs to divide. Before we spend any energy on division, first we want to make sure that the cell is capable of division. So we're going to check the DNA. We're going to make sure there isn't any damage to the DNA. If there is, if the cell can still survive and do its function, it'll be kept. If it's not, a process called apoptosis is going to occur, which we'll talk about in a little bit, which is programmed cell death, and that cell is going to be removed. If everything is okay at G1, then we go into S. And at the end of S, S, we're replicating the DNA. At the end of S, proteins are going to check the DNA to make sure that it replicated correctly, that there weren't any mistakes. If there are, then that cell is going to be destroyed. Once you get to S, you can't go back to G1. And so if something's wrong, apoptosis kicks in and we destroy the cell. At the end of G2, there's another checkpoint. Here we're making sure that, again, we're checking the DNA, but we're also making sure that we're ready to build a spindle, that we've got all the extra nutrients, the extra monomers, polymers, organelles that's going to take for one cell to become two. And if everything's okay, then we go into mitosis and start prophase. And the final checkpoint is in metaphase, where we're going to make sure that first the spindle's fully built, but second, that every pair of sister chromatids are attached to a separate spindle fiber, and that those chromosomes are all aligned down the middle of the cell. If all of those things are true and are okay, then the cell goes ahead and starts anaphase and finishes mitosis. And this is a really hot topic. The cell cycle checkpoints and how they function, how cells are controlled in mitosis is a really hot area of research in biology. And the main reason for that is because of cancer. And in the next lecture, we're going to see how cancer is specifically related to the cell cycle. And so there's a lot of money being thrown out there right now for research on this area. Okay. So again, G1 checkpoint, we're making sure that there's enough, there's a need. There's a need that the cell's big enough, it's matured enough to grow. We're checking the DNA so it's not damaged. Anything that any of those aren't met, the cell's not going to be allowed to go into S. If it's not allowed to go into S, we can either stop the cycle and try to fix it, or it just goes into G0 and wait for signs and conditions to be better, or apoptosis occurs, programmed cell death. In the S checkpoint, again, we're checking to make sure all the DNA got replicated correctly. If not, apoptosis. And what happens in apoptosis is other cells will identify that this cell is damaged and needs to be removed. And so cells have a death receptor, and it's going to bind a signal molecule, and that causes this process called blebbing to occur. You don't need to know that, but this is the name for it. See, we don't want to just lyse this cell and release the cellular contents, because many of those cellular contents are chemicals called cytokines, which will trigger an inflammatory response. And that inflammatory response can be a big problem. A lot of diseases, the inflammatory response is worse than the actual damage caused by the organ. And COVID-19 is like that. For people who have extreme COVID, it's not the virus, the effects of the virus that are really causing all the problem. It's the inflammation that's happening body-wide. Ebola is the same way. Pyogenic E. coli infections are like that. Clostridium difficile infection of the intestines is like that. So what we do instead is the cell blebs, and what it's doing is it's breaking into smaller pieces, each surrounded by membrane. And so the membrane's keeping those cytokines inside. Now our macrophages can come up and can phagocytize these and get this cell out of our system. Okay, and that's apoptosis, programmed cell death. Again, the G2 checkpoint, this is where we want to be sure we're big enough. We've got all the reserves of proteins, but also carbohydrates and lipids, organelles, extra membrane. We've broken down the cytoskeleton. And again, we're going to check the DNA and make sure it's still good. And again, if any problems are detected, we either try to fix it or apoptosis. And then finally, the M checkpoint, toward the end of metaphase, when we're sure that all sister chromatids are correctly attached to a single spindle microtubule. And then the cycle isn't going to proceed until the kinetochores and their proteins in each pair of sister chromatids are firmly anchored to at least 2 spindle fibers, one from each side. Okay, and if this doesn't happen, then a process called non-disjunction can occur. We'll talk about this in the lecture when we talk of meiosis. But basically, disjunction is when the sister chromatids separate. So non-disjunction is when they don't, and both go to one daughter cell, and nothing goes to the other. And there are some issues with non-disjunction that are probably the biggest one is Down syndrome. But we'll talk about that in a future lecture. There are also some intracellular molecules that regulate the cell cycle. Some of these are positive regulators that either induce cell growth or promote movement to the next step of the cell cycle. The more important of these that you're going to read about in your textbook are cyclins, and then other molecules called cyclin-dependent kinases. Negative regulators stop the cell cycle and stop advancement. Retinoblastoma protein, typically just called RB, and P53 and P21 proteins are the big three in this. And we'll look at these in a lot more detail in the next lecture when we look at how mitosis can lead to cancer, or how the two are related. And oftentimes it's when these molecules have mutated so that they can't do their job anymore. And that's the end of this part of Chapter 10. Here's the terms that you're going to want to study for the exam. Be sure you know the definition of what they're a part of. And then these are the performance objectives, the key concepts you need to know in order to do well on the exam.